together. Maybe you're wondering how this bread related for our topic today. One of the main ingredients of this bread is what exactly we are going to talk about. Do you have any guess? Well, if you're thinking of yeast, you're absolutely right. But before going to our experiment, let me give a little details of what are yeasts. Yeast is most commonly used in the kitchen to make dough rise. Have you ever watched pizza crust or a loaf of bread swell in the oven? Yeast makes the dough expand. But what is yeast exactly and how does it work? Yeasts are eukaryotic single-celled microorganisms classified as members of the fungus kingdom. Yeast, any of about 1,500 species of single-celled fungi, most of which are in the phylum Ascomycota. Only a few being are in Basidiomycota. Yeasts are found worldwide in soils and on plant surfaces, and are especially abundant in sugary mediums such as flower nectar and fruits. The main difference between yeast and bacteria is that yeast is a eukaryote whereas bacteria are prokaryotes. Further, yeast has a single nucleus per cell, but bacteria do not have a nucleus. Yeast and bacteria are unicellular organisms. The materials we will need are 3 glass cups, 2 teaspoons of sugar, water, warm and cold, 3 small dishes, pen or marker, ruler, stopwatch or cell phone timer, and of course, yeasts. But before we go to our experiment, let's have first our hypothesis. In glass 1, if we mix warm water, sugar, and yeast, we will produce a foam-like bubbles. In glass 2, if we mix only warm water and yeast, nothing will happen. And in glass 3, with just yeast on it, also, there will no changes at all. So these are the procedures. Fill all 3 dishes with about 2 cm of cold water. Place your glasses in each dish and label them 1, 2, and 3. In glass 1, mix 1 teaspoon of yeast, 1/4 cup of warm water, and 2 teaspoons of sugar. In glass 2, mix 1 teaspoon of yeast with 1/4 cup of warm water. In glass 3, place 1 teaspoon of yeast in the glass. Now start your timer. After 10 minutes, stop and measure the height of the froth in all 3 flasks using a ruler. Repeat the experiment 3 to 5 times to establish reliability of results.
A few moments later.
So these are the data we gathered. In setup 1, its appearance looks frothy and foamy. It smells like spoiled food and sour. Its texture is sticky and slippery. In setup 2, its appearance is liquidy with bubbles. It smells like spoiled food and sour. Its texture is slippery. In setup 3, no changes. It smells like sour. Its texture is rough and cold. In cup 1, the height of the froth in trial 1 is 2.5 centimeters, in trial 2, 1.5 centimeters, in trial 3, 1.7 centimeters, in average, 1.9 centimeters. In cup 2, trial 1 and 2 got 0.5 centimeters, in trial 3, 0.3 centimeters, in average 0.4 centimeters. In cup 3, all of the trials have 0 centimeters. There is a foamy substance in cup 1 and 2, but cup 1 has more froth than the second one, while the third one stays the same. Nothing happens. Cup 1 had more yeast foam because of the presence of sugar that is converted to glucose to enable it to feed into respiration, and it is fermentation process causes the foaming. Our activity proves how sugar affects the yeast growth. So the more sugar there is, the more active the yeast will be, and the faster its growth. I am very optimistic before doing this activity. Challenge accepted. I know at the end of this activity, I will learn much about the yeast. It makes me excited in such an experiment like this. While doing this activity, the foaming stage of the mixed sugar, yeast, and water makes me more fascinating as I repeat the procedures 3 times. The challenging moments comes when the smells of the setups gets more stink as I repeat it more times that possibly happens because of the yeast. Learning moments come as I repeat the procedures and measures the froth and compare the setups. Specifically, I learned that yeast may smell badly but they are substantially beneficial to human culture, in particular for the production of breads, alcohols, and other foods.